Hello everyone, welcome to Module 8, Study Closeout. This is a 2-week module. The first week we're going to look at study closeout activities, essentially what you need to do to shut down the database and to finalize it for your project. The second week we are going to have our midterm exam. I'm going to be posting the date for that soon, as well as a review lecture. And I will also be hosting a review WebEx. I'll send you the invite for that as well. So this week you need to watch this lecture, of course, and look through all of the assigned readings and videos that I have posted. Please let me know if you have any questions.
So I know this overview slide looks like we're going to cover about 1 million things. And we are going to touch on each of these, but very briefly. Again, we don't have a red cap assignment for this module, so basically I'm going to discuss the typical locking and database closeout activities, and then you're going to be getting a lot of information from your readings. So we're going to talk about both soft and hard locks or freezes. We'll also look at the reasons why we might soft lock a database, for example, an interim analysis or a safety monitoring review. PROSCA says a locked database defines the point at which final analysis can start and conclusions can be drawn on the data. And that's well and good, but what do we mean when we say we have locked the database? Well, in most cases, that simply means we've altered the settings on either individual user accounts or the data sets themselves to ensure that records can no longer be changed in any way. They can't be modified, they can't be deleted, you can't add to them. Why would we want to do this? Why would we want to remove access to these records and stop changes? Well, we do it because we want to preserve a snapshot of the data that was used to create an analysis. So for example, if you ran your analysis on 50 patients worth of data and you knew that at that point in time they had 70 adverse events, if somebody wanted to look a little deeper into the adverse event profile and wanted to run new programs to review it, you would want them to use exactly the same data that was used to produce the first analysis. So we actually are going to lock down the database, and we create data sets typically and transfer them out into another directory where we lock access to those two. There's a lot of locking, a lot of access restriction. There are generally two types of locks. A hard and soft lock. A hard lock is intended to be final and permanent. You usually do this at the end of the study, and the intention is that you're going to freeze the database forever and ever at the point of analysis. And a soft lock, however, is intended to be temporary. You use a soft lock when you want to stop updates for a short period of time while you take a look at data that has been collected to that point, or perhaps data that meets certain criteria. For example, you might want to look at all patients that have gone through dosing regimen 5. So I wanted to point out, you can also hear people use the term freeze instead of lock, so don't be surprised if you hear people talking about a soft freeze or a hard freeze. Some people distinguish between the two, but really I think they're used pretty interchangeably in the industry. So I mentioned in the previous slide that before you lock the database, you need to make sure you have all the data and that you have done everything to it to make sure it is complete and reliable and valid. So there are a number of steps that you typically go through, and at the end of this we'll talk about the concept of having a checklist, and PROSCA has some nice checklist examples in the readings that I've signed to you. But one of the first things you do is make sure you have all the data in the database. You don't want to be, sorry, there's a siren going. You don't want to be missing any pages of data that perhaps were left at the site or not collected or not filled out. You don't want to be missing any eCRF screens. You want to make sure that the site entered all the data for every patient they have. And in particular, you want to know that you have all the critical variable data that was produced for the study. So how do you figure this out? Well, typically you have tracking systems, whether they're paper-based or electronic, and those tracking systems will compare a patient who completed per protocol to all the patient data that you get, and it will tell you if there is data missing, and then you have to determine whether it's validly missing, for example, the patient discontinued, or whether discontinued early, or whether it is data that you need to go after and retrieve. You also want to reconcile your CRF database to any other data that you might have received for the study outside of that database. A very common reconciliation task is to compare the serious adverse events to the adverse events. And what you're looking for there is to make certain that for every adverse event that is indicated as being serious in your database, that it was actually reported appropriately and entered in the serious adverse event database, which is the safety database. Another very common type of reconciliation is when you get data imported in from a central laboratory. So in other words, you don't enter that data as a case report form screen, but you get a set of data sets on a regular basis from the central lab. You then have to compare your patient and visit records in your database with the laboratory samples, dates and visits and patient identifiers, and make certain that for every patient you have a corresponding lab for all visits, and for every lab you actually have a patient in your database. So you do that reconciliation both ways. You also need to make sure all your QC checks are complete before you lock a database. With a paper-based system, typically, we've done some of these now. You guys did a great job. You're going to compare your CRF to a listing of data from the database. You are going to look at a random sample of patients and review 100% of their data to make sure your overall procedures worked. And I know this gets confusing because I'm using 100% twice, but you look at 100% of the data for a randomly selected sample of patients. However, you typically look at the key variable data for every patient in your database. So you look at 100% of the key variable data. In an ED system, life gets a little easier. Primarily, you're looking at query resolution to database checks. Sometimes you'll pull a sample to make sure that you think to just look at the overall data and make sure the site followed the data entry or CRF completion guidelines. But your big focus is really on the query resolution process. You have to make sure the primary investigator signed the case report forms. So I have this little note here that to comply with the requirement to maintain accurate case histories, clinical investigators should review and electronically sign the completed eCRF for each subject before the data are archived or submitted to the FDA. Use of electronic signatures must comply with Part 11. They mean 21 CFR Part 11. So in most cases, you have to have the PI sign them before you archive the study. But it also they have to be signed before the results are submitted to the FDA. And a lot of companies just want to see that it's signed before they begin their analysis. And that's going to really be something that's done on a company-by-company basis. But remember, at some point before you close out your trial, whether it's before the lock or after, you've got to make sure that the PI has e-signed every form or physically signed the paper forms, and you have to give a copy back to the site. So once you've completed all the steps to make sure that your database is complete and accurate and valid, you're going to go in and actually lock the records. So I mentioned earlier you can have different options in different databases for the level you lock to. For example, you might be able to lock down access to all data. Or you might be able to access data or lock down data access for specific fields or forms or patients or investigative sites. It's very common to want to lock by site because you get your CRAs out and do your closeout visit, and then you lock that site and nothing else is going to change in it, and you can go on and close out the other sites. In REDCap, we don't have as many options, primarily because REDCap's not meant really to be used at a large number of commercial or big sites. But what it does allow you to do is lock down the entire record for a patient or lock it down by form. So you can see I locked down the records for the forms for record number 1, patient number 1, and I did not lock down the visit 3 data, but I locked down all the other data. And over on the right, you can see that I had 2 options. I could lock all forms, or I could unlock all forms. So you have a few options in REDCap, and most systems are like that. You just need to find out what your options are before you start to lock. So since you have a lot of steps you need to go through before you lock your database, you'll want to have a checklist, and typically that's going to be a study-specific document, and you're going to store it in your data management folder when you're done, or your data management study file. There are a lot of different tasks and steps that are going to occur, and many of them will be project-specific. For example, if you do not have a central lab, you don't have to do the central lab to CRF database reconciliation. So you need to look at all the moving parts of a particular project and then create your project-specific lock list. And I have an example here that's just from, I think, one of the hospital-based sites, but you can see some of the things that they look at. This is for paper studies where they have to make certain that all the CRFs have been collected and that all the data clarification forms or queries are resolved. So those sorts of things. This type of plan is typically just titled a database lock checklist or checklist. And I encourage you, when you do your reading, to examine Proxka's 2 examples. She has them in Chapter 13, Study Database Lock, which is your assigned reading. They are figures 3.1 and figures, or 13.1 and 13.2. So enjoy that. That will be very helpful. After you've locked the database and your primary investigator has signed all the forms, you need to archive your study. And there are actually some regulatory requirements around that. And there can also be sponsor-specific requirements. But the primary thing is that you are going to have to retain, or the sponsor and the site will have to retain the records for a period of time. And this is in case there turns out to be a problem with the product or questions about the results of the trial. The regulatory agencies want to be able to go back and reproduce the documents that were used to create the database. So preparing the study for archival, if you've kept your data management study file up to date, you really are only going to need to add the final documentation of the locking that you did. If you did not keep your data management study file up to date, I will be very disappointed with you if I find out. Then also you're going to need to take some time and file your project-specific documents, and then you have to transfer them to the trial master file, and that's the main file of essential documents that you are probably studying about now in CLR430. Remember that you need to provide the site with a copy of the final CRFs. Section 4 of ICHE 6 requires that the site retain a copy of the subject records for 2 years after the last marketing application, that's a submission to a regulatory agency to market the drug, or the development of the drug is discontinued. So if you stop a drug because it's not working, you still have to retain the records for 2 years. You can print a copy of the eCRFs, or you can burn them onto a CD, and if you have paper CRFs, you can send those, or the site will keep those at the site. So study archival. Make sure you keep all those essential documents. So why would we need to do a soft lock? We've talked about a hard lock, and that seems to make perfect sense. You've ended the study, you're done, you're going to file everything. Why would we want to do a soft lock? Well, often we do them because we're doing some sort of analysis in the middle of the study, whether that's focused on efficacy or whether that's focused on safety, or even trying to identify a minimum therapeutic dose or a toxic dose. You stop the trial in the middle of the study and you take a peek at the data and you see what it tells you. So a couple of things we want to talk about. An interim analysis is something basically you conduct before all the patients complete a study. Like so many other elements of a clinical trial, you need to plan your interim analysis before the study starts and before you begin the analysis. So you can't start poking around in the data and say, oh, this is what I want to look for, these are going to be my criteria. You have to define all the criteria of your analysis in advance. You usually have an interim analysis plan that details what will be reviewed and when it will happen. And the trigger for that analysis is often when a certain number of patients reach a critical phase in the study. So for example, you might not want to look at your interim analysis data until 50% of the patients have been enrolled and completed phase 1. Generally, you're going to freeze all activity in the database, at least for a short period of time, while you transfer the data sets out or extract the data and send that data to the analysis group. Once the interim data sets are archived and safe, generally we can go back and work in the database. Sometimes they'll ask you to stay out of it for a couple of days or a couple of weeks. That's very rare, but sometimes they'll say just stay out of the data. We don't want any changes until we are done with the interim. Why do we do them? Why do we do this interim analysis? Well, generally, we're taking a look at the safety and efficacy of the trial. And there's two reasons. First, actually there's a variety of these reasons, and one of the assignments you have is to go through an excellent presentation by a gentleman named Murph on data and safety monitoring and clinical trials, so I'm not going to try to speak too much of that. But in general, you're looking to make sure there aren't any safety issues with the trial. Also, you want to make sure that the drug hasn't shown such strong efficacy that it would be unethical to continue the trial while some patients were assigned to placebo or comparator treatment groups that didn't work as well as the study drug. So we often think a lot about the fact that we stop a trial if it's unsafe, but we also stop it early if it works. So this is just my reminder slide that I want you to carefully review the Data and Safety Monitoring in Clinical Trials PowerPoint produced by Harvey Murph. He does not narrate it, so you're going to have to read through the slides and pick the information out in there, but I think you're going to find it worthwhile. I was in the industry for years before I understood the criteria for an interim analysis. I knew we did them, I knew what I needed to do, but I didn't fully understand why or what the outcomes were. So I want you to have that information because it will help you understand how to do your job better. When you do look through the Murph presentation, you're going to see he discusses why what the factors are that we look at in an interim decision. And I think you'll be surprised because, as I said before, it's not just safety. They also check recruitment rate and completion schedule. And the reason they do this is if the patients aren't being enrolled quick enough, you might say, we're never going to get to the sample size we need, and therefore, again, it's unethical to have the patients we have enrolled continue with the trial. You might look at the baseline characteristics and then assess the risk profile, kind of a safety issue. You could compare baseline and make sure that you actually have a patient population and an appropriate control. You could see how well people at the sites are complying with the investigational product as an intervention treatment. You might assess whether the data is complete and we've got appropriate follow-up for the patients. You might be looking at internal consistency. So what are the outcomes and what is the safety profile? And you also just might be looking at statistical issues. Do you think that you're getting your, again, sample size and power, and also ethical issues. So there's a lot of things that people might look at when they take that interim peek at the data.
So what are the requirements for when you do a DSMB versus when you don't need to do one? Well, here, particularly in the U.S., the two major groups that are overseeing our research are the NIH and the FDA. And the NIH usually does require a DSMB if there are protocols that are going to be using blinded or randomized data, if there are protocols with more than one site or center, and for gene transfer protocols. They might require a DSMB if in general, the trial poses a greater than minimal risk to the patient population, or protocols that require special scrutiny, such as those that have a really high public interest. For example, think about Ebola recently, or that are being run in vulnerable populations. For the FDA, they are really recommending a DSMB when there is a lot of risk to trial participants based on either the study endpoint or whether it's a particularly large, long-running trial, or if the population is fragile or at risk. Sometimes they do it for practicality in a short trial. And also, they are looking for a DSMB when they really need to make sure that they can assure their scientific validity to the trial. So this happens a lot when you're working with novel drugs or doing something in a novel indication. They want to make sure that there is no bias.
Another very common reason to do a soft lock on the database and send a snapshot of the data out is to have somebody perform some independent monitoring. So, a lot of the review that is done on a trial is done by a CRO hired by a sponsor or by a sponsor, but there is a requirement for many trials that the safety and efficacy of the data is reviewed at interim points throughout the trial, key points, by a group that is totally independent, that has nothing to gain from the drug being approved or the trial continuing. And so there's a variety of different independent monitoring groups, and I just want to give you a few of the more common names. You might hear people talk about a data and safety monitoring plan. That's essentially a plan that says how you're going to monitor the progress of the trial. What are you going to check, and how are you going to ensure the safety of the patients? And what is the mechanism for reporting adverse events to the IRB and for ensuring data accuracy and protocol compliance? So that's sort of, the DSMP is typically utilized by a DSMB, a Data and Safety Monitoring Board. And again, this is a group of people that are qualified to look at the results of the trial and determine whether there are safety signals or, in some cases, efficacy signals that would require the trial to be stopped early or changed. You might also have a clinical endpoint or events committee. So, for example, if you're doing a trial on, let's say, this is a very simple one, probably not the best example, but if you're doing a trial and you want to ensure that patients genuinely had a heart attack, that they just didn't have symptoms that appeared to be myocardial-like, then you might have 2 or 3 independent reviewers review the laboratory results or the EKG results for patients that are entered in the trial, and those 3 independent reviewers would review that event and tell you whether or not it was actually a heart attack or something else. So again, you might hear people talk about a CEC. And finally, another term is Independent Data Monitoring Committee, and this is really a lot like a DSMB. The terminology is just a little different. And again, you're seeing, I hope, a theme here that the focus, all these words say safety or monitoring or event, and really you're looking at having an independent group that's taking a look at the data and saying, are these patients doing okay, and are we running this trial all right? So, do we do or use DSMBs or independent monitoring committees for every trial? No. The NIH and the FDA actually have some hard requirements for when you need to have that independent monitoring. But and sometimes sponsors will decide they want it anyway, because if something goes wrong with the trial, it's nice if the sponsor can say, we weren't hiding anything. We had an independent group review this. So some sponsors do it even if they're not required to. But Murph will talk more about this, but in general, the first decision tree is it clinical research or not? If it's not clinical research, generally you don't need a DSMB. If it is, then you want to ask yourself, is it a phase 3 trial, which is typically larger and more involved, or a multi-center trial? If it's phase 3, you're usually going to want to have a IRB and a data safety monitoring plan and a data safety monitoring board. If it is a multi-center trial, then it's still up in the air. Maybe you need one, maybe you don't. So maybe you have a phase 2, but you have a lot of sites. So then you have to ask yourself, is the trial blinded? Is the intervention or product that you're using high risk as opposed to low risk? Or are you in a vulnerable population, let's say people with diminished capacity? If the answer to any of those are yes, then you're probably going to need an IRB and a data safety monitoring plan or a data safety monitoring board to go with that plan. If the answer to all those questions are no, and your study's not blinded, there's not a high risk in the intervention you're using, and your population's not vulnerable, then no, you don't need to have that data safety monitoring board. So I think you'll enjoy reading through the Murph presentation. It's good information. I think you're going to feel strange because there's not a whole lot to do with this part 1. No assignments in REDCap. Really, you just need to review all the materials that I have in Module 8. I promise I will use some of them on the midterm exam. So, you know, I put this material in there because it's going to enhance your understanding of the lecture. So make sure you do it. Read the assigned Proxca text sections. I will have those listed. And, you know, some of these, the chapters and pages I know are a little wonky, so look for the chapter headings and section headings. That will help you navigate no matter what page number you have for your version. Make sure you read the articles, and please look over Murph's PowerPoint in the content folder. As always, call me if you have any questions, or email me if you have any questions. And I will be posting a review PowerPoint next week that's going to cover the major key topics from the first 8 modules, and should help you prep for your exam. Good luck.